Yeah, all right. Good. So thank you everyone for coming for our next talk in the CHIP speaker series. I'm excited to introduce Dr. Rishi Kamaleswaran. Yeah, close. Rishi is an associate professor at Duke University. He has appointments in surgery and anesthesiology, as well as biomedical engineering and electrical and computer engineering. So lots of appointments. He's also director of the informatics core at the Duke Clinical and Translational Science Institute, similar to NC Trans. A computer scientist by training, Rishi explores ways to use machine learning to improve patient outcomes. His research aims to develop intelligent systems that can be used to develop new cures for diseases and to advance clinical understanding of critical and acute illness. So I'm excited to hear his talk today about his work on explainable real-time prediction in the ICU context. Thank you so much, David.
Thank you again for the invitation to host me here. I'm so excited to meet you all and to present the work that we do in human critical care ethics. So I'll start off by obviously introducing the concept or the motivation for why we think that machine learning and artificial intelligence has the opportunity to significantly change how we care for the health of critically ill patients in the ICU. Typically, patients in the ICU are they arrive to the ICU for a number of reasons. Some of them can be acute injury. It can be something like a car accident or infection. Others come to the ICU for complications that occur during routine procedure, like a surgical procedure that just became complex, and so the patient now had to be stabilized and then cared for in a very complex way. But ultimately, they come to the ICU with a complex physiology, a complex biology, and there's a lot of things that are going on. And essentially, clinicians are trying to stabilize these patients and bring them back to health. This broad notion of salutogenesis, so this shift towards a healthy state. But what often happens is that even the most trained human, the most trained clinician will miss things that occur basically below the surface in a sense for these patients. Some things that we call, for instance, hidden hypoxia. You know, sometimes they may have a very low oxygen concentration of maybe hovering just above that 89%, but never really triggering an alarm for desaturations, but they may be persistently below that range. And that's concerning, especially because that may ultimately result in a seismic event like a cardiac arrest or a code or something that occurs down the line, which clinicians maybe because of this highly dynamic and busy environment, they often lose track of. And so there's this notion that data-driven techniques can allow for earlier identification of these very complex interactions. So what I mentioned was just simple in a sense, a single unimodal change. But what may happen, for example, is that there may be an associated shift in the heart rate, a shift in the oxygen level in a way that just seems like abnormal. And so if someone is constantly exposed to signals all the time, they may miss out on those salient divergences. And so there's really an opportunity for us to capture that complex change. What's really also a good opportunity is that unlike other fields of medicine like cancer or other kind of endocrinology or other things, critical care is still in a very syndromic phase of medicine, which syndromic generally means a symptom. It's a constellation of phenotypic changes that appear to be, this or that. It's kind of like, the shade of yellow. Like it's like, how do you tell what is yellow? You kind of guess or estimate that the degree of yellow is this or that, but it's really syndromic. So sepsis, an area that I study and I'll introduce a little bit more, is a hallmark of syndromic conditions. sepsis is by design characterized as a dysfunctional host response, host meaning the human response to an infection, which is dysfunctional. That means that the normal response that they should see in most humans, for some reason, this person is responding in a very abnormal way, maybe too aggressively, maybe not aggressively enough. So, but the degree of response can lead to a constellation of changes. So it can lead to organ injury. It can lead to increased what we call tachypnea, or this increased breathing rates. But that's just like the superficial things that we observe. That's not really tied to the infection per se. It's a response to that infection. So syndromic in that sense means some things that we see changing, not really tied to the underlying etiology, but just kind of a symptom of that underlying etiology. And sometimes that's really hard to know. It's hard to know when someone comes in. Are they septic? Do they have an infection? Autoimmune? Are they like, is it an allergenic response to something? Or is it something like an internal bleeding, or like, there's a constellation of things which have the same syndromic manifestations, which are very different in etiology. So the way to respond to those things can be really complicated. And so we think that there's actually an opportunity, once you can provide a very a data-driven approach to characterize these changes, there's an opportunity to better identify what that underlying etiology is. Why is this patient here in distress, so that proper treatments and interventions can be applied. The second, and I think huge opportunity, some of those provocative stats, right? So sepsis has been studied since humans existed. I mean, it's been an evolutionary part of our existence to respond to these pathogens that are constantly trying to kill us. But we still do not understand why, given two people, two 25-year-old individuals, why one person decides to have a significant response and why someone else has a weakened response. Is it because this person came from a privileged lifestyle, that they were naive in terms of their immune exposure, that they had not seen as many pathogens as someone that came from a more disadvantaged environment, that has a huge repertoire of adaptive immune response? Is it that, or is it that the degree of the pathogen, the insult was so significant and not equal amongst the two? Or is it that the response to the infection was unequal? There's so many factors that we have to try to characterize. And so we think that by utilizing methods like graph-based methods and causal inference, we can try to characterize beyond the phenotype what is causing someone to respond in a maladaptive way, and look at underlying etiologies, and then look at mechanisms, and then look at novel cures or novel treatments. That's to say, sepsis has been studied again for decades and decades. There's been 100 clinical trials that have been addressed to, or that have been funded by the NIH to address sepsis. Zero success, right? So this is huge investment, billions of dollars in investment, zero success. So this is still an unanswered question. We still don't know. The complexity is significantly higher than what we can currently use, or foresee models or other things. Because how do you replicate the ability of the human and the injury that they have in a very controlled experimental model? You can do like single ligation punctures. You can try to tweak a little bit and then see what the response is. But that's not how humans get to the ICU. But that's not what happens. So we need new human-based models to study this disease or this syndrome. And that's an opportunity that we have. And that's where we think that data-driven methods and techniques can help us. So I discussed earlier why someone shows up to the ICU can be complex, but how they show up is fairly consistent. So this is a really nice figure. It kind of shows you that as people start from a um, 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 how they start. So for example, if they start in that blue phase, that's that phase of health. They move through that phase of health, let's call that baseline. They get some injury. And then they start to progress through that phase of injury into this area that called the island of opportunity. Let's just call that white area, the island of opportunity. They're still not in distress, but it's not obvious to anyone looking at them. There's no phenotypic signs. But then, you know, the infection brews and the acute injury begins to get significant enough that it crosses into that threshold of detection, where people now start to see, oh, wow, this person looks pretty sick. They're like, you know, breathing fast, their heart rate is going high, their fever is going up, they seem delirious, they don't know what's going on. And then they progressively get worse and worse and worse. So the questions that we've all had in our minds is, if we had a predictive algorithm that could be invoked earlier than when humans detect this condition, could we reverse injury? And what we often find is the yellow line, we identify something as it occurs, or maybe just a threshold of occurring. We reverse something, but then they never really return back to their baseline health. They have some persistent organ injury, they go home with a need for lifetime of dialysis because they have consistent renal dysfunction, or they go on with some constant dementia. It's a huge problem in the ICU. Because they're like loaded with drugs, with sedatives, with paralytics, with all sorts of things, that there's a neurologic change to that. And really our opportunity for changing the paradigm of critical care is by identifying these manifestations before it happens. But the grand challenge is, how do you know someone is sick if you don't see that, or if you don't detect it? So this is where we've been looking into, this is where the biology really comes in. So the notion is it a genotypic tendency? Are we born with an increased proximity, or are we just more likely to develop sepsis as a condition? One thing that we do know from the Danish twin study, or sorry, the Icelandic twin study, is that if you were a male and your parents had died of an infection, the chance that you would die of an infection is up to 5 times as much. So there seems to be something in there, but is it hereditary? Is it a combination? Is it that you just are likely to, but then there has to be these additional factors that come into play? And the American population is not like the Icelandic population. Like it's just, there's just so many more fires and smokes in there. So what causes all this complexity? And this is where I think the opportunity is for novel discoveries, and continues to present opportunities. But once they're in the ICU, let's say we detect them, we know that they're in distress, we know that they're just at that baseline of that threshold. How do we prevent them from going further? Because what happens is when they show up to the ICU, they look like they're okay, but you don't know whether this person is going to be in the ICU for 2 days, or whether they're going to be there for 7 days or 14 days or a month, right? You don't know yet, because that first 24 hours is going to tell you whether this person is, but in those first few hours, it's hard to know, because they all look the same. So one of the things that we've been looking at is there's a lot of data that's being collected and that's being amassed in the ICU. We have waveforms, we have physiologic data, we have huge millions of variables that are being collected. And so we've been looking at one of those data points, the physiologic data streams. And we've been capturing a lot of this data, and we've been characterizing these subtle changes in physiology to see, can we phenotype or can we cluster these groups to characterize who is likely to stay for more than 24 hours, have multi-organ dysfunction, have complex clinical course, versus the group that's likely to be mild and then get back to health. And our questions are, what's the host resilience factor? What keeps someone, what's internal to them that allows them to rebound quickly and get back to their healthy state, versus like this just absent and just progressively the house just the roof catches on fire and then progressively everything gets worse. So unfortunately, the reality is that in many of the ICUs, while we're collecting this data, we're actually not storing the data, and we're downsampling it into a couple of vital signs that we put into our EHR records. So actually, there's millions of data points that are being generated, are downsampled down to like one heart rate, one sun rate, one respiration, one blood pressure, one temperature. Right, so all of this data, maybe every 2 hours at most, right? So the hypothesis that we've been really studying is this link between what we call the autonomic nervous system, or the brain, and our ability of the brain to control the different organ systems. The brain is essentially responding all the time to stimulus, to environments. It's controlling our heart rate. If we're in this room, we're relaxed, and suddenly a tiger jumps out and everyone's heart rate starts to go up because the brain detects something and it starts to do its thing. And so we've been looking at this autonomic nervous system, and we've been trying to see what is the autonomic nervous system doing in patients that have a very poor response to infection and have multi-organ dysfunction and so forth. And what we've shown over and over and over again is that response is pretty poor, and you can separate them. And there's been lots of studies, and this has been studied for decades. People have started as early as the 1990s, have shown that there are these biological oscillations, and these oscillations seem to be abnormal, and it's been nonlinear. But we've seldom had methods to really characterize and tease out what those differences are. So what we've started doing is we've started looking at each of these signals in different cohorts of population. So initially we started looking at these signals in adults. We captured all this data. We looked at individually every single data stream, and we said, what do these streams look like as they progressively get septic? And what we found was that as early as 24 hours before clinicians get suspicions of infection, which means that they order blood culture or they administer antibiotics, there are these changes in heart rate. And there's these changes in what we call altered heart rate indices. But then somewhere in the middle, there's a shift from a cardiovascular to a respiratory or pulmonary shift. And this is a cohort that we're very interested in, because this is a cohort where they quickly, their lungs quickly fill up with a lot of phlegm, blood, water, edema, all this kind of stuff, essentially preventing them from being able to breathe by themselves. So they need ventilatory support and they need aggressive therapy. But not all patients go down this path. So it's a subtype. And the question is, why is it that this subtype is at an elevated risk for pulmonary complications? And so we've been looking at this is work that we started doing at Georgia Tech, where we started building these acoustic microphone sensors, where we would put on patients, and we did this in a controlled setting in patients with heart failure. But in heart failure, it's similar that there's this backfill of fluids, and these fluids start to dump into the lungs, and then you get edema, and then they start to get worse, and then you have to put them on diuretics to kind of pee out that excess water to get their lungs to breathe, to breathe back again. But the same sort of thing occurs in patients with sepsis, except it's not necessarily water, it's just the inflammatory, you know, the mess that starts the debris, the dead cells, like all of this stuff that started to build up, which then promotes further debris and further inflammation. So we're now moving towards, you know, looking at lung acoustics along with our continuous signals to see, you know, can we track the lung physiology over time? And this is what you're seeing here is like a spectral map of each breath that the patient's inhaling. And during that time, we're able to characterize those patients who develop these crackles and these wheezing type of markers to see what the depth of edema they have. So we can kind of see where localized that edema is, and then how it's growing over time and manifesting. So this kind of gives us a real-time view into the lung complications over time. And you know, this is again a preliminary study that we did in heart failure, but this is you know, can directly transfer into the ICU. The problem in the ICU is that unlike in heart failure, about 30 to 40% in the medical ICU are on mechanical ventilation. So in addition to all the sounds of the lungs, there's also this big machine that's just constantly out there, which eliminates a lot of the acoustic signals that we see. So we're now trying to see, okay, can we try to, you know, look at bioimpedance and we try to combine those. So it's still an ongoing problem that I think needs further investigation as we move from the spontaneous breathing population to the mechanically ventilated population. Another aspect that we're also characterizing is something that we call hemodynamic stability. So because we're collecting all of these signals, we're also interested in non-invasive markers, because it's seldom that you actually get an arterial line put in. Maybe about 20% of patients in the ICU have a continuous monitoring of their blood pressure. But a hallmark of sepsis is septic shock. And septic shock means a drop in blood pressure, in mean arterial pressure, to a point where there's not enough sufficient perfusion to the rest of the organs. So one of the primary reasons why patients with sepsis go on to have a complicated ICU stay is that in the mess that occurs, that inflammatory response, their vascular tone starts to get very weak, so that you get vasodilation, which we think of a river during a heavy rain. There's a lot of water going through, but during drought, there's very little water. So if there's very little water and the red cells can't move from one place to another, then they start to, the organs that depend on blood flow for oxygen gets compromised. And so then you have ischemic injury to end organs like your liver, your kidneys, and the spleen. So two organs that have a very hard time dealing with this is the kidney, number one, and the brain. The kidneys, the amount of AKI or acute kidney injury that we see in this cohort is amazing. It's a devastating complication that occurs because the kidneys they always need to be perfused. Similarly, the brain, the brain can tolerate oxygenation, but it has a lot of levers it can do to push more oxygen to the brain and at the expense of other organs. So there's all this stuff that's going on. So one thing that we're interested in is, can we identify the patients that have these complex cases, these complex vascular response, and can we try to do it non-invasively so that we don't have an actual mechanical pressure sensor that's being inserted that potentially opens them up for a secondary bacterial infection, which we would like to avoid. And we've been trying to build this towards a non-invasive blood pressure monitoring program. And so we use these markers called pulse arrival time, which is basically, if you look at the EKG, the first beat of the heart detected as an heartbeat on the electrocardiogram to when the pulse peak is detected on the pulse oximeter. It tells you how long, it took the blood flow to reach from the heart down to the fingertip. And that's a proxy of vasomotor tone, or how strong your vascular tone is. And so we've been using this and other instantaneous markers to estimate what the mean arterial pressure is. And so we did this, recently in a study, using data from the Emory ICUs to show that just by looking at just 4 variables, the most recent mean arterial pressure, which is taken by the cuff, not by the invasive mechanical sensor, but looking at the cuff map, and then looking at what that pulse arrival time metric is, and combining that with instantaneous heart rate and heart rate variability, you can actually build a very robust model to predict septic shock. And we did this, in 6 hours before vasopressors were initiated, all the way up to 36 hours. And also, although you get different degrees of predictive performance, you can start to see these variables, have a shift. So this is an example of a patient that was in the ICU, where you're seeing there in the top, in the second line graph, mean arterial pressure. Again, take note that that line, that dotted line, is the map of 65, at which point anything below that is recommended for interventionists to treat. And you can see that this patient is generally, stables, and this is hours before. They're stable for some time, until then, over like a couple of, or more than 24 hours, they start to really kind of go fluctuating up and down, up and down. And then finally, they have a crash right at that 70th mark. And that's a map of less than 55, which is actually, for some people, they start to go into a coma, anything below that, because they can't tolerate, the brain can't tolerate a drop in oxygen, so it just starts to shut down. So similarly, you have, other fluctuation of the markers that are changing. And in the same case, you have this one, but, unlike these markers, what you're actually seeing here is the amount of changes in pulse arrival time, which again starts to drop lower. You start seeing changes in inflection index of blood pressure. And again, all these markers show that there's increased change. And then what do you see clinically? Clinically, when you're monitoring these things, it's through this measure called lactate. Lactate is if you ever go to the gym or you run, you start to build lactic acid as you get more anaerobic respiration going on. And as your cells' oxygen demand goes up, but the supply is not there. So the cells start to use glucose and go through glycolysis. That means that you're really not in a suitable state, that the demand is so high that the available oxygen is not sufficient to go through the Krebs cycle and generate more ATP. So you start to see a rise in lactate, right? When that supply demands problem starts to come up. And so this it was very late in the process, right? So there's other opportunities that we have. So relying on what we know to be biased, so relying on a metric where we detect it after a problem has become so catastrophic that the host biology has to shift its entire energy consumption structure to deal with something, is too late. Is I think not really. So there's, again, this is where we think that there's an opportunity for us to utilize these and make better sense. So I do want to point out that it's not all the right answers. It's complicated because, as I mentioned earlier, we're not just waiting and watching these people respond to the sepsis course by themselves. The humans are actively intervening to arrest their deterioration and to bring them back from that state of rest. So what we notice here is, we built this really nice indicator using protein variability and other things to separate between sepsis and COVID-19 to see if we characterize what is a bacterial infection versus what is a single virus, right? If we can differentiate the two, be golden. We never have to give antibiotics if someone shows up with a viral infection. Why waste our time? We can address the viral pathway independently. But then, what we start to notice is that when we look at these patterns, especially if we're using retrospective data, the noise that is introduced by these interventions, especially mechanical ventilation, has an ability to modify physiology. So how our body responds to breathing and heart rate is through these very, very sensitive receptors or reflexes. And there's especially one right in our throat called the baroreflex node, which is responsible for how we regulate respiratory and hemodynamic status. So as you start to have these like massaging or these manipulations of breathing rates, it changes the way that the brain adapts to physiology because it thinks that something is normal or abnormal. So it then changes, what we start to see in terms of cardiac rhythm is also a little bit shifted. So this is something that we start to notice in these patients. The other thing is we've been looking at a lot is this notion of temporal transitions. Not everyone stays in one physiologic state from the time they come in to when they leave. They in fact change, and what we show is that you can actually project these physiologic states in up to 28 different state clusters, states in their state, and they can move back and forth between those states through their ICU course. So the question is there like a motif or a symbolic representation of the transition states or transition probabilities, which it tells us about what type of injury they have and what type of progress they're going to. So what we did is just did like a basic NLP type of analyses. We took each of these states and we said, okay, like run like a word to back type of transition state. So if we see like a trigram or like A to C to F, and then we plot those and we cluster them in a k-means. And then what we generated was that there could be distinct cohorts or clusters that we can derive from this trigram, and it separated those acute phases. So there's some phenotypes which are strongly associated to death or complicated course, and there are other states which are not so strongly associated. So from a binary division standpoint, we could see that there are these changes, but this is just scratching the surface. I think there's just a lot more that could be done to better separate these phenotypic states, to better understand what is the organ of injury. Is it kidney? Is it liver? Is it heart? Is it brain? And then how do you respond, and what interventions worked? What promoted someone to a certain state? What kind of prevented them from going to a certain state? We also started looking at some of these endotypes or these underlying biologic states. So we do a lot of work with omics and genomics data. So we are often taking blood from these patients because we can't take tissue, we can't do anything more. We look at what are the endotypic states, and because we can only see what's in the blood, we can't go to the organ where the infection is and kind of see what's happening there. We see a lot of response, immunologic response. And what you're seeing here is when we look at these patients with sepsis, you're seeing a markedly aggressive neutrophilic response, right? So the way that we evolved, right? When we evolved through generations, is we have this innate immune arm and we have this adaptive immune arm. The innate immune arm is what we are given through our parents. Like if we inherit that. When you're breastfed, you get certain types of antibodies which grow your innate immune arm. This is the most primitive part of our immune system. This is what we inherited through evolution. The adaptive immune arm is what we learn as humans in our lifetime. This is the distinct marker that characterizes us. There's this whole domain of immune naivety. You know, when you grow from a privileged background, you have a very naive immunity versus if you saw an infection every other week, right? Then your adaptive immune cells are just much stronger. Challenge is if they're too strong, that's a problem. If they're not strong enough, that's a problem. So there's all these things that we don't appreciate. But what you're seeing, that big red blob in the middle is our primitive immune arm that is aggressively, dominating the landscape of immune cells. And we've known that neutrophils, think of neutrophils as your suicide bombers. They kind of go in when they sniff out something coming in, they go in and they just blow up. And when they blow up, they damage everything. They damage your lungs, your tissues, everything around it. Our evolutionary tactic to seeing DNA in the blood has been chaos. DNA in the blood, that is not where DNA should be. It should be right in the nucleus inside cells. So if I see DNA in the blood, I freak out, I call other things and I say, attack that thing, remove that thing. But the people that come first are these macrophages and neutrophils, because the adaptive arm hasn't woken up yet. It needs about maybe a couple of patients who have naive immunity to create those antibodies, to create those helper T cells to kind of go in. But what we show here is that what we find is that there's an aggressive immune response that starts almost immediately, and it's going there to try and get rid of these pathogens. But by looking at just the blood, it's hard to say. You can say, you can create these two groups, ones that survive, the ones who don't have an aggressive response, and the ones that do have an aggressive response. That's good, but again, not necessarily helpful to know what should you do as a clinician. Okay, I understand that this is someone that's going to die. How do I help them, right? That's the first question. So what we've been looking at is we've been looking at this data and then applying those phenotypic characterization, the physiologic characterization to say, okay, you have an aggressive response, but this person seems to have a lung-related complication. And so we've been characterizing them and we've been able to show that when you do apply that phenotypic layer, you can actually get even more granularity. You can separate amongst those two endotypes. You can separate them into 4 different groups where one is really, really sick and you need to throw the kitchen sink at this one because you don't want this person's out. Versus someone else who seems very aggressive, but you may have a bit more time. So you may be a little bit more judicious in how you respond. So there's all these, I think, opportunities, and this is just, again, scratching the surface. The other things that we've been also working and playing a lot with is, I think, the great opportunity in simulations and generative AI and all the things that come on, is to take our immunologic data and start simulating what can happen if I target this pathway. What is the associated change in the immune landscape? Can I lower the immune cells? Can I try to make that precision intervention? So we've been using a lot of these open source libraries to simulate what happens when you knock out a certain gene and knock out a certain pathway. But it's a complicated process, and this is where I think there's a lot that needs to be done. The challenge is that the methods and models and causal structures have fundamental assumptions in temporality, which is not the case in the data that we deal with. A lot of our data is Bayesian in nature because they're sampled with a very strong a priori assumption. But then they're also Markovian in a sense, because I don't care about how you got to this state. You got to this state, I have to work with what I have. So there's a lot of opportunities for new methodological developments to characterize this very complex structure to better understand what are the fundamental components. So we've been developing a lot of these Bayesian causal models and trying to look for what happens in irregular regular sample time series versus regular sample time series. We've been using Hawkspin processes to try to project the irregular, like the more aligned irregularity. And we've been seeing some benefit, but again, it's telling us what we already know. Antibiotics reduce infections. This is fluids resolve shock. Like things that we know, so we still have some ways to go before we start to better understand how to move in terms of an actionable, informative causal structure. So in the last little bit, I just want to point you to another aspect which I think is a great opportunity in this field for those that are really interested in pursuing this. While I talk to you about a lot of physiology, a lot of biology, this is we live in a privileged environment. We live in an environment where we start to, if you have sepsis, the chance that you're going to die is about 10%. If you get to their ICU. But in areas of the world where the infrastructure doesn't exist, if you have sepsis, the chance that you may die is up to 90%. Right, it is a death sentence. So what can we develop in terms of methods, interventions, and tools that can allow for us to identify these concepts earlier? And so we've been moving towards this notion of like assessing our the quality of care that patients receive. So this is an example of what healthcare is today, right? Grandma has a fever from a nursing home, ambulance drives to the nursing home, picks up grandma, brings her to the emergency room. She gets, looked at, maybe goes to the ICU, maybe gets some interventions. Then everything resolves. We put her back into the ambulance, send her back to the nursing home, and then again, we wait for this to happen in the future. So this notion of a centralized, let's respond as we get issues has been what's dominated us in the post-World War II era. But for thousands of years, medicine has been practiced at home. When you were sick, people came to see you, and you got treated, and you got intervened at home. So our vision for the future of critical care management is what would the ICU look like if it could be done at home, right? If it could be done in a decentralized way. And this is, aligns with a lot of recent calls from national agencies that have been looking at or really asking us to reimagine the future of healthcare because these centralized models have now become, now they've started to work so well that the cost of these models have become so expensive and unsustainable that there's an economic need to try to provide cheaper and economic alternatives that are decentralized in a sense. And also the rural healthcare in America looks pretty bad right now. Hospitals are closing left and right. So there is a need, another urgent demand. So we've been thinking about what does it look like for a decentralized ICU, and we're starting to explore some of these concepts in terms of support systems and so forth. So this is like ARPA-H's, I don't even know if ARPA-H exists anymore, but their vision of what the future of the decentralized ICU or the decentralized healthcare looks like, where, rather than going to the hospital to get, this is a true story, right? If you have end-stage renal dysfunction and your kidneys don't work, you need dialysis, you need hemodialysis. And you need it about 3 times a week. So most people don't have time to go to the dialysis clinic, sit there for three hours or whatever it takes 3 times a week. I mean, that's a lot to ask. So people try to maximize. They try to regulate how long they try to build some internal resistance, and they try to say, okay, I'm gonna go twice a week this week, and then I'm gonna go once a week. But it's really stressing. Imagine you don't pee for a week, like you only pee once a week. Imagine what that would do to us, right? So So this whole notion of complexity is brought upon these new models where, could we actually have this dialysis machine go to you, like drive to you, and then you get your dialysis at home, and then it goes to the next person and so on. So similar like this, we're starting to think about what does this look like for the ICU. And if you think about what is an ICU, it's respiratory management, it's a ventilator, it's renal management, managing the kidneys, primarily, and then hemodynamic support, managing blood pressure. And then everything else is added, right? So crucially, you're managing the lungs, the kidney, and blood circulation. So could we get to that point? So we've been looking at like what are the minimal needs to try to sustain that. And so this is our view of what that would be combining vision with some interactive language to help remote intensivists see what's going on and manage that, built within a robust, resilient site infrastructure. And that's, I think, where we could actually start having some of these interventions being done at home, or even if it's in the field by the partner, or it's like, say you're hiking the bottom of the Grand Canyons and someone, has dehydration and then goes into shock, and you start resuscitating them by, infusing fluids. So a lot of these things are not, they're not Herculean, they're not complex, but they need to be done efficiently. And there's opportunities to provide these to those people. So in closing, I'll just say our approach to how we have sought to study this very complex sepsis course, which if anything, I tell you, there's a lot of hype and there's a lot of news in the media that sepsis has been solved in 2018, or like, that it's not a condition that's worth studying. I would beg you to reconsider, because I think we have only scratched the surface of understanding the very complex host response, our biological response, and there's miles to go in how we build better treatment, better interventions, better therapeutics, better strategies for managing patients. So there's a lot to be done, and we're welcoming all sorts of collaborations and partnerships. So thanks so much for listening. I want to thank my lab, who's been a huge part of this work, and also thank the funding agencies who've supported us so far. Thank you so much.
Thanks for a great talk. Any questions from folks? Well, I certainly have one. So I guess it's something that's been floating in my mind, and then it came up very directly at the end of your talk, where you had the ARPA-H vision of the truck coming around, and you mentioned disaster areas. So I recently had a conversation about disaster response and telehealth. So in the case, not necessarily where a truck is going to drive around, but maybe that will happen. But we now have all kinds of devices that measure all sorts of different things, and I wonder what the opportunity is for the same kinds of things you're doing, but there's flooding in North Carolina and no one can get to hospitals. Can we let people download an app that will help them take care of a parent or somebody and know where to go with the truck, right, or the helicopter to help people?
I think that is, I would even push that a little bit further and say, most of our ICUs are probably just hoteling spaces for people that are just staying there because we don't know what if, then something happens. So maybe, we can utilize these wearable sensing or even if not wearable sensing, even, have these newer alternative health models where some of our lower trained healthcare individuals can accompany them and can provide those services under the guidance of a more experienced individual. But I think this is the opportunity for computing in this space. How do we how do we facilitate that asynchronous communication better so that people know that this has been done, that's been done, rather than still trying to go through, Zoom calls, and what telemedicine today is, I think not what people really envisioned telemedicine would be, maybe when they started talking about it 20 years ago. It's moved from, let's have sensors on and see everything and see nice dashboards to, let's put on Zoom and kind of get your laptop on, right? So I think there is a huge opportunity, and as we start to really think about, I think, maybe what's happening in this current administration or this current is going to force us to rethink about what we do and what patients hospitals care for, and maybe shift that earlier or even outside of hospital.
And the other thing that, just in that same context, is as the cost of going to the ICU is enormous, right? So if you think from just the cost perspective, as we learn what signals are predictive and informative, I can imagine it's not just the devices we already have, but maybe there's some low-cost kit.
Yeah.
Like we have emergency kits for hurricanes and other things, where people can have those devices around.
Yeah.
And maybe they're measuring things we aren't measuring now, but they're things that are clinically really informative.
Exactly. Yeah, I mean, just like the COVID test kits, right? If we could have a test kit for whether you have a bacterial infection or not, or, is your lactate rising, right? And so, or even like a blood pressure sensor. What's your blood pressure now? It could be really small.
Any other questions?
For the presentation, on the more technical side, I know you talked about historical data and sound data. Was there any point in this study that you incorporated them together? Maybe like one on Multi-Modal, and how difficult is that?
It's a great question. I think it's always, with these, it's challenging to find like patients where you have all of them together. Because it's complicated to collect blood and to capture sensors, and so what we've been doing is we've been looking at phenotypes. So we've been looking at like groups. So we've been seeing, okay, if you, with some of the biologic data, if you exist in this cluster, this cohort, with some minimal amount of clinical data, we reproduce that in the other domain. We try to actually through kind of combinations, but getting the depth of data in those is, unless you have a controlled clinical trial where you're actually, prospectively monitoring, it's like using retrospective data is a huge challenge. That's not to say it shouldn't, like I think everyone should be capturing these data, but actually it's only a handful of hospitals in the country that capture this type of data. Thank you so much. Thank you for